The overall goal of this procedure is to observe patterns of gene expression in zebrafish embryos during specific stages of development. This is accomplished by first hybridizing a digoxygenin-labeled antisense riboprobe to the mRNA of interest. The second step is to detect the riboprobe using an antibody for digoxygenin. The antibody is conjugated to an alkaline phosphatase enzyme. The third step of the procedure is staining by the addition of NBT and BCIP, which react with the alkaline phosphatase enzyme to yield an insoluble purple precipitate. The final step of the procedure is to incubate sufficiently stained embryos in methanol to reduce non-specific staining. Ultimately, results that show gene expression patterns in whole zebrafish embryos at specific stages of development can be obtained using whole mount in situ hybridization.
The main advantage of this technique over existing methods, like traditional classroom demonstrations of histology or dissections, is that it gives students the opportunity to build conceptual bridges between molecular biology and trends in organismal development and evolution, thereby providing a truly integrative experience.
Although these methods have traditionally been used to study the development of a few model organisms, they can easily be applied to other non-model organisms to understand the conservation of developmental pathways across vertebrate taxa, as well as where these pathways deviate, leading to evolutionary novelties.
This method may be unfamiliar to undergraduates, so some students may struggle at first. But learning this method will serve them well in undergraduate research experiences and upper division courses.